Hey everyone, welcome to The Explainer. Today we're exploring a truly fascinating study that was recently published in Urologic Oncology. We're going to zoom way in to look at a microscopic battleground inside the human body, specifically right inside the bladder. We're looking at how a localized ecosystem of bacteria interacts with our own genetics to either fight off or stealthily aid a really relentless cancer.
All right, before we get into the weeds, let's clear something up. For decades, medical science operated under this assumption that the human bladder in a healthy person was a completely sterile environment. But is it? Well, the short answer is absolutely not. Modern sequencing has totally busted this myth wide open. The bladder actually hosts a complex, thriving ecosystem of bacteria, kind of like our gut. And when that ecosystem gets thrown out of balance, things can go wrong, very wrong.
Okay. So here's our roadmap for decoding this really dense, multi-layered study. We're going to cover 1. The NMIBC recurrence problem. 2. Meet the bladder oncobiome. 3. Transcriptomics and the matrix. 4. Good bacteria versus bad bacteria. 5. The microbial gene network. And finally, 6. The future of personalized medicine.
Diving right into Section 1, the NMIBC recurrence problem, a persistent threat.
So, NMIBC, that stands for Non-Muscle Invasive Bladder Cancer. And it's a huge deal, making up about 75 to 80% of urothelial carcinomas. Now, on paper, the early prognosis for this type of cancer sounds relatively favorable, right? Because it hasn't actually invaded the deep muscle of the bladder wall yet. But here is the massive catch. It is notorious for returning. You can surgically resect it, completely treat it, and yet this cancer just keeps coming back like a relentless invader. The big question researchers wanted to answer was simply why? What exactly is making this local environment so welcoming to a recurrence?
Which brings us to Section 2. Meet the bladder oncobiome, the local populace.
Okay, so to really grasp what's going on, we got to define a crucial term here, the oncobiome. In the context of this study, the oncobiome refers to an alteration in the mucosal microbiota. That's the bacteria lining the bladder. We're comparing that to the healthy tissue right next to it. Think of it as the tumor's neighborhood. And what we see in this neighborhood is a massive drop in the diversity of species, plus a big shift in who is actually living there. And the visual proof of this altered neighborhood is just striking. If you look at the alpha diversity, specifically the species richness, notice how the data points just absolutely plummet when you move from the normal tissue on the left over to the abnormal tumor tissue on the right. The tumor tissues exhibit a statistically significant reduction in microbial species richness. Think about it like this. A healthy bladder mucosa is kind of like a bustling, diverse rainforest. A diverse population is a strong, resilient one. The tumor environment, though, it's completely lost that diversity.
Moving right along to Section 3, Transcriptomics and the Matrix, the fortress walls.
So, to figure out what was happening to this physical fortress, the researchers used something called shotgun meta-transcriptomics. Essentially, they looked at the RNA to see exactly which genes were being turned on or off. They mapped all this out using a volcano plot, which is basically our translator for this microscopic crime scene. Every single dot represents a gene. The ones pushed way up into the far left or far right, those are the genes behaving super abnormally in the tumor tissue compared to the healthy tissue.
Let's break those complex dots down into some simple numbers. The researchers identified 57 total differentially expressed genes, or DEGs, but look at the balance here, it's wild. Only 12 genes were upregulated, which are those red dots on the right, but a massive 45 genes were downregulated, shown by the green dots on the left. Think about what that actually means. The tumor environment is triggering a massive, coordinated shutdown of specific genetic instructions.
So the absolutely crucial point here is, what do those 45 shutdown genes actually do? Well, when researchers ran a gene ontology analysis, they found that the vast majority of these downregulated genes govern the extracellular matrix, or the ECM. The ECM is basically the biological scaffolding, the structural concrete of the tissue. But turning off these genes, the tumor environment is literally dissolving the structural integrity of the bladder. The fortress walls are weakening, which completely rolls out the red carpet for the tumor to expand and easily return.
This perfectly sets up Section 4, good bacteria versus bad bacteria, guards and saboteurs.
Because, those crumbling walls aren't just crumbling in a vacuum. The study identified a super clear clash between some specific microscopic players. On one side, we've got a pro-tumorigenic saboteur, Propionactobacterium acnes, or P. acnes. On the flip side, we have a protective guard,
Bifidobacterium longum. Let's see how they match up in the tumor environment versus healthy tissue.
First up, our saboteur, P. acnes. Now, you might actually recognize that name from skin acne, but it is causing way deeper problems here. In normal bladder tissue, it makes up about 13.3% of the microbial population. But look at the tumors. It completely spikes to nearly 24%. No way, right? P. acnes is known to increase inflammatory markers, so it's essentially acting as a saboteur, creating this pro-inflammatory microenvironment that cancer absolutely loves.
Now, contrast that with our protective guard, Bifidobacterium longum. This specific bacteria is widely recognized as an anti-tumor mediator. It maintains tissue homeostasis and boosts the immune response. In the normal tissue right next to the tumor, it's sitting at almost 4%, but right where the tumor actually forms, it is severely depleted. We're talking crashing down to less than 1%. The guards have basically been wiped out.
And that takes us to Section 5, the microbial gene network, the battleground map.
So how do these bacteria and the host's genes actually communicate? Are they just occupying the same space, or are they really talking to each other? Well, it turns out they are highly interactive. Researchers mapped this entire battleground, and it resulted in this crazy complex network detailing 80 statistically significant associations between the microbes and the host genes. The squares represent biological processes, the circles are our genes, and the diamonds represent the bacterial species. It's a seriously tangled web of communication.
Let's go ahead and decode the really key connections here. Genes like SBS-SPON and TMEM119 act as these central hubs. And we see certain bacteria, like Vita-Spar and C. durum, strongly interacting with genes that regulate matrix deposition. But the most important thing? Look at our hero guard, B. longum. The network shows it is directly linked to TMEM119 and FBN2. And what do those genes do? They regulate the extracellular matrix. So B. longum is actively trying to interact with the structural genes to maintain those fortress walls. But remember what we literally just saw? B. longum is severely depleted and those exact ECM genes are shut down. The host's entire defense network is failing.
Let's move into our final section, the future of personalized medicine, rebuilding the walls.
So what do we actually do with this map of a failing fortress? Well, the study's authors conclude with a pretty powerful statement. They say, these host-microbe interactions may contribute to disease pathogenesis and recurrence. Further studies are warranted to elucidate the underlying mechanisms and therapeutic implications. This beautifully summarizes our whole exploration today. It's not just the cancer cells acting all alone. The loss of structural integrity in the bladder wall, combined with an invasion of bad bacterial actors like P. acnes, plus the loss of good guards like B. longum, it creates the absolute perfect storm for this cancer to keep returning.
Which leaves us with this really provocative final thought. If a dysregulated oncobiome is helping the cancer recur, what happens if we fix the neighborhood? If we can map a patient's specific bladder microbiome, could we one day prevent cancer recurrence not just with chemo or surgery, but by introducing targeted probiotics? Could we literally rebuild the fortress walls by simply bringing back the right bacteria? It is a fascinating frontier in personalized medicine, and for sure, one we're going to be watching very closely. Thanks so much for joining me on this explainer, and keep asking those big questions.